Hello students, we are discussing about the life cycle of lambda bacteriophage. So we know that the lambda bacteriophage has got two cycles. One is lytic cycle, another one is lysogeny cycle. So these lytic and lysogeny are regulated by a host of regulatory elements. We discussed in the previous video the function of N, CRO, C2, C3, O, P and Q. The relative amount of these regulatory factors will decide whether the lambda phage should enter into lysogeny cycle. Another one is the lytic cycle. In recombinant DNA technology, we are not interested the lambda bacteriophage enter into the lysogeny cycle, but we are interested the lambda bacteriophage to enter into the lytic cycle. So now in case of a lytic cycle, we are going to study the various aspects of lytic cycle under different headings. First one is going to be the immediate early transcription, followed by delayed early transcription. After that, we are going to have the DNA replication. In the DNA replication, we studied in the previous video that the early replication is bidirectional, so that we do have the circular lambda DNA is going to be synthesized. Then later, the DNA replication is converted into rolling circle model in which a tandem repeat of lambda DNA is going to be synthesized, which is very critical for the packaging. Now, this particular video, we will discuss about the late transcription process. So now this is describing the late transcription as well as DNA packaging. Once the DNA replication is over, the next step is going to be the synthesis of various proteins responsible for the assembly of head as well as the assembly of tail region, in fact. So here what happens, the head is synthesized separately and tail is going to be synthesized separately. Then into the head, the DNA is going to be packaged in, and finally the tail is going to assemble, so that we are going to have the final active lambda bacteriophage. Now what this particular figure represents is that they have initiated the lytic cycle and they have stopped at different time points. Try to understand what are the stages of assembly with respect to the head as well as with respect to the tail region. What I mean to say that they will initiate late transcription. So now during the late transcriptions, it is transcribed into mRNA followed by the protein. Now these proteins, how these proteins are going to be assembled. So they have done it at, okay, they have started the lytic cycle and they have stopped the reaction in the very early. Then they only found out all the head and tail proteins are present in the form of a soluble proteins. They are not even assembled yet. Then later what they have done it up, they have stopped even a little bit later period. Then they try to characterize what happened to the soluble proteins. Then these soluble proteins are all assembled and then they have identified a particular precursor, what they call this as a scaffolded prehead. Then further, when they incubated for a further longer period, then they identified one more stage, what is called as a prehead. So like that, they have identified the various stages involved in the late transcription. So we are going to study how the soluble proteins are going to be formed and what are the assembly particles which we can characterize, and how these assembly particles will be processed so that DNA is going to be packaged, in fact. So earlier what happens, they have identified that all of them are all present in the soluble form, but the earliest precursor they could identify and characterize is going to be the scaffolded prehead. Alright, what I have shown is a schematic representation of the various particles which they have characterized. Now here, this scaffolded prehead means I think all these proteins are assembled and this is the earliest precursor they could identify, and this has got lot of proteins are assembled, and this particular scaffolded prehead is not convenient for DNA due to packaging. So now what is required is that some of the additional proteins which are packaged has to be removed. The scaffold has to be removed, Now the removal of some additional proteins, what they call this as a processing of this pre-head, is done by a host protease called, derived from the gene GROE. So GROE is a gene. Now the gene product of GROE is nothing but a protease. Now what this protease does is that the extra proteins which have assembled are all going to be cleaved, what we known as the processing of scaffolded pre-head. Now that removal of these additional scaffold proteins results in the formation of another important stage. We call this as a pre-head. Alright. So now from the scaffolded pre-head to pre-head requires a proteolytic cleavage. So you must be wondering that a protease inhibitors are excellent inhibitors for as an antiviral drug, because most of the viruses will also have a lytic cycle. So in that lytic cycle, so they are going to have an assembly and then they have to process further with the help of a protease. So most of the protease inhibitors which are specific acting on such proteases turn out to be a very good antiviral, because if it is not processed, then the virus particles is not going to be formed. So that is very important. So from scaffolded pre-head to the pre-head, we need a proteolytic action. Now that is from the GROE gene product, what is known as a host protease. So scaffolded pre-head will be converted into a pre-head. Now in the pre-head, I think we do have a space in the head. So in this head, I think we are going to package the DNA. For that, we also discussed that it has to be a tandem repeat of lambda DNA because it requires 2 cos site. Now the next step is with the function of a NU1 gene product and A gene product of the lambda bacteriophage. What these NU1 and A does is that it will bind to the cos site. I remember that, I think, because we do have 2 cos site. So now they will bind to the cos site and then they will attach to the mouth region of the pre-head, in fact. So this is the important step. So now that one is carried out by the NU1 gene protein, another one is A gene protein, in fact. What they do that they will specifically bind to the 2 cos site and then they will bring these 2 cos site very close to the mouth of the pre-head. So that is the major function of NU1 and A. Next stage what they have identified is that with the action of F1 gene product, the DNA which is going to be incorporated into the empty head. So now this incorporation, or whatever the packaging we call it, is carried out by the function of F1. Alright. So now during this particular time, what happens? The pre-head will be increased by 20% and again they will they will attain a particular shape like an hexagonal. Now here it is a circular, and because of the F1 action, when the DNA is going to be packaged into the empty head, so now the size of the particle increases by 20% and again the size will also attain a definite shape in the form of a hexagonal. Alright. So now the next step is going to be that from this step to this step, so what happens is that there will be an addition of certain protein, what is called as a D protein. So what happens is that D proteins will get assembled on this empty head. So now they are called this as a D protein also because of it kind of a decoration which is given to this particular head. They are also called as a decorative protein, in fact. If these particular proteins are not assembled, again the particle is not viable. So this is a very important step. So now the D proteins are going to be assembled. So once it is assembled, now the shape of this particular head is fixed. So now that is going to be an hexagonal. So now it is a scaffolded pre-head. So now proteases are going to be processed this scaffolded into a pre-head. It is also called as a maturation step. So now we have the pre-head. So into this pre-head, with the help of NU1 and A, the 2 cos sites of a tandem repeat DNA is going to be attached to the mouth of the pre-head. Then with the help of an FN, it is going to be packaged in. Then the size is increases by 20% and attain a hexagonal shape. Then to this hexagonal shape, D proteins are going to be assembled on the surface. So now they are called as a decorative protein, and this protein is very critical. So that will ensure the shape of the size and fixes the size as the shape as an hexagonal. Now what happens is that A is always assembled at the cos side. Now the A has got a endonuclease activity. We call this as a TER function. So because of the endonuclease activity of A, which is attached to this one, it will cleave at the cos site. Alright. Now the remaining DNA is going to be removed. So now as a result, now we are going to have a linear double helical DNA structure. So since it has been cleaved at the two cos side, so we are going to have the 12 base pair overhang. Alright. So now that is because of the TER function. So now this is the packaging of the head. The next one is got independently. So now the tail is going to be synthesized. Now the gene product which are involved in the tail is J, I, L, K, G, H, M and U. So all these proteins assemble to form a tail. I told you that head is synthesized separately and tail is going to be synthesized separately. Now with the help of F2 gene, W and Z gene product, the head and the tail are going to be assembled. Alright. Now this is how in case of a late transcription, proteins are synthesized and they are going to be assembled and processed. While processing, the DNA is going to be packaged in. Then after that there are some more additional processing of DNA. Then that gives a definite shape of a head which is hexagonal. Then the packaged DNA is going to be cleaved. Then tail is going to be synthesized separately and the whole particle is going to be synthesized. So now once the number of particles increases within the host system, then the last step is going to be the lysis. Now the lysis is carried out by the gene R as well as S in fact. Suppose like if you do have a mutation, for example in the S gene, then this particular system fails to hydrolyze the E. coli cell membrane and it is unable to come out of the host cell. So now the whole process of mutating S genes so that the number of virus particle is going to be increased within the host cell. Since there is a mutation, how are you going to get this particular particle? By mechanically or using detergent lyse the E. coli cells so that we can get the whole particle. So a mutation of this particular S, we do have an advantage so that we can get more number of phage particles in the host system. Now this is about the late transcription as well as DNA packaging and the various steps involved in the process in fact. The next one we will discuss about the lysogeny. So before we get into the lysogeny, I just wanted to bring my previous one. So in case of a lysogeny, the important one is that the repressor gene C1 has to be elevated. So now for the elevation of the C1, the C2 which is a positive modulator is also going to be elevated. So because of that, this HFL which is a host protease specifically cleaves the C2 has to be inhibited. So now that one is carried out by the C3. So now the C3 concentration should also be increased in fact. So once the C1 which is a repressor gene is elevated, what it does is that it will bind to the both PL as well as to the PR and stops, it only works up to immediate early transcription but not delayed early transcription. So at the elevated level of C1 during the lysogeny cycle, there is an INT protein is also increased as well as a little bit of XIS gene product. So now what is the major function of an INT protein? what INT protein does is that both, so now this is the forged DNA and this is the host DNA. So now the forged DNA has to integrate into the host region. So what this INT protein does is that it recognizes an ATT site both in the host chromosome as well as in the lambda DNA in fact. So it recognizes specifically the ATT sites, and this INT will initiate cleavage. So as a result, so we are going to have the opening of this particular host DNA as well as this particular lambda DNA. Now after cleaving that, now then there will be a joining of this end with this end and this end with this end. Now that results in the integration of the host, the lambda DNA into the host genome. Now the lysogeny is not lethal to the host because the host undergoes multiplication as the host genomic DNA multiplies. Along with that, even the lambda DNA also multiplies. So there is no harm. So now this is going to be integrated into the host DNA. That is what is known as a lysogeny cycle. But now how are we converting the lysogeny into the lytic cycle? So now that again depending upon the host repressor gene, that is the C1 repressor gene, has to be removed from the binding in fact. So I will just discuss in detail about what exactly is a C1 protein. C1 protein is a repressor protein consisting of 236 amino acid. What they have also found out that, so now they also found out a temperature sensitive mutation. What do you mean by temperature sensitive mutation? Because this C1 protein is functionally active at 37 degrees centigrade, so that C1 is going to be synthesized in large amount. Then INT proteins are going to be synthesized. There will be an AT8, there will be an ATT. So then the lambda DNA is going to be incorporated. But once the temperature has been increased from 37 degree to 45 degrees centigrade, then the C1 repressor protein becomes inactivated. So now that is the inactivation occurs at 45 degrees centigrade. Once the inactivation occurs, then the C1 repressor protein is no more functional. Now at that particular time what happens? An other additional gene, that is an XIS, is going to be synthesized in large number. What this XIS does is that now there is an ATT site here and there is another ATT site here. So now this XIS joins these two ATT sites. Like we are going to get the original genomic DNA and excising the lambda DNA. So now to convert lysogeny into lytic cycle, somehow we have to unblock the C1 repressor gene. The moment we have the C1 repressor gene is unblocked, then another protein, that is an XIS protein is going to be synthesized in large amount. What this XIS does is that it will join the two ATT sites in the host genomic DNA. So by this joining with this particular site, so now we are restoring the original genomic DNA, and this particular lambda DNA is going to be excised. Since there is a lower concentration of C1 repressor gene, so then we are going to have the delayed early transcription followed by DNA replication and the late transcriptions, and that results in the lytic cycle. So now here I just want to, in this particular slide, I have just given lot more information on the various type of promoters which are used in the recombinant DNA technology. And so now we do have promoters of antibiotic resistant gene. Then we also have a little bit advantageous promoter, that is going to be the laxi DNA. I will explain. So compared to the laxi DNA, the C1 repressor protein is still more advantageous. I will explain why and how. So now in case of the plasmid DNA, I think we discussed about the common antibiotic resistance which are used. So one is the ampicillin resistant gene, tetracycline resistant gene, chloramphenicol resistant gene. Another one is a kanamycin, which is a aminoglycoside. So ampicillin resistant gene, what happens like the ampicillin antibiotic, what it does is that it will inhibits the various enzymes involved in the synthesis of cell wall, and that's how it kills the microorganism. In case of a tetracycline, they bind to the 30S subunit of the ribosome and blocks the protein translation, and that's how it acts its antibiotic activity. On the other hand, chloramphenicol binds to the 50S ribosome subunit, and again it inhibits the protein synthesis and kills the microorganism. So whereas kanamycin again binds to the ribosomal subunit and interferes in the protein synthesis and kills the microorganism. But these microorganisms also developed resistance. How do they develop? So in case of an ampicillin resistant gene, they produce an enzyme lactamase and hydrolyzes the beta-lactam ring of the ampicillin so that it will make ampicillin ineffective. So there is an enzyme. So if you want to synthesize this particular enzyme, the ampicillin resistant gene should have a promoter. Alright. Another one is going to be the tetracycline resistant gene. So this resistant gene is again because of a protein. What this protein does is that it will make the membrane not permeable for tetracycline to enter in. So now this is again a protein, requires a promoter. So in case of an chloramphenicol, now this chloramphenicol synthesizes an enzyme. So now this particular enzyme together with acetyl-CoA derivatized chloramphenicol to hydroxyacetoxy derivative. So as a result, the derived chloramphenicol unable to bind to 50S subunit so that the protein synthesis is not blocked. So whereas in case of kanamycin resistant gene, again it produces an enzyme aminoglycoside phosphate transferase. So now this enzyme again modifies the kanamycin and it is unable to bind to the ribosome. So what I want to tell you that all of them offers resistance because of the synthesis of proteins which interfere in their antibiotic action. So that means all of them for sure do have promoters. But these promoters are not in anybody else's control. The moment you have this ampicillin resistant gene, so it is going to be expressed immediately. So compared to the promoters of ampicillin or tetracycline, LAGZ DNA is advantageous basically because even though you have the LAGZ DNA, the LAGZ DNA is not going to be expressed because this activity is dependent on the lactose concentration. If there is a high concentration of lactose, then the LAGZ DNA is going to be functional. If the concentration of lactose is low, LAGZ DNA is not functional. So as a result, even if you do have the LAGZ DNA, the promoter is in our control so that we can really regulate. So and we also discussed in detail that we don't use the lactose as a positive modulator, but we use an analog. So now that is IPTG, isopropyl thiogalactoside, which is an analog. So if you just add this IPTG, so then we can initiate the LAGZ DNA. But here it is not in our control, but here we have to include IPTG. So you just include IPTG into the medium, so we have the function. But the advantage of C1 repressor protein over LAGZ DNA basically because here the promoter is temperature sensitive. We don't have to add anything. All we have to do that we just change the temperature of the incubation medium. So we don't, because here we have to add an IPTG, but here we don't have to add anything. So the C1 repressor protein is easily under our control and we don't have to add anything. All we have to do that we just increase the temperature. So if you want this C1 protein along with this promoter, so we can always at 37 degrees centigrade now all the cells will get into the lysogeny cycle. Okay, now if you want to convert them into the lytic cycle, so we don't have to add anything. So for its activation, all we have to do that increase the temperatures to 45 degrees centigrade. So this the promoter choice is very important because we are going to study this in our future vector design. So I will stop at this particular point. Let's see how you people have followed the whole process. Thank you very much.